Hello everyone, very good evening to all. So we have started our second chapter and which is the microorganism, friend and foe. In that chapter, we have already completed the microscope, what are microorganisms and the types of microorganisms we have completed. We have gone through about the like bacteria, protozoa, fungi, algae and viruses. Now today we are going to start the useful microorganism. So first we have to understand what is their useful in our body or for the human welfare, that we have to understand. So in milk we have that lactobacilli bacteria. See, bacilli word is used there, that means they are rod shape bacteria. So lactobacilli bacteria which is present in the milk, it is present in the milk. So in milk we have, milk is sweet in taste, right? Milk is being sweet in taste because it contains lactose sugar. So I'm writing here milk
So it
contains
contains lactic acid also.
lactose sugar. Lactose sugar. That's why milk is sweet in taste. It is sweet in taste. But in milk we have lactobacilli bacteria, and lactobacilli bacteria have capacity to convert that lactose sugar into lactic acid. Lactic acid. And due to action of lactic acid, what is there in milk? So in milk we have water and lactose sugar and protein substance. Okay, we have lactose sugar, we have water and protein substance. So due to action of lactic acid, the protein substance is being separated out from the water. This process is called as coagulation, and milk turns into the curd. Milk turns into the curd, and curd being sour to taste. Why it is being sour to taste? Because lactic acid is there, and acids are substances which are sour to taste. Okay, so milk is being converted into curd, and its flavor also like changes. It changes from sweet to sour. Okay, it is due to the lactose sugar converted into lactic acid. So here that lactobacilli bacteria, lactobacilli bacteria are responsible for that process. Okay, and these lactobacilli bacteria are very useful because they used to kill harmful microorganism or harmful bacteria which used to grow in our alimentary canal. Okay, like we have clostridium. Clostridium is a harmful bacteria which causes food poisoning, and lactobacilli bacteria used to kill those clostridium, means harmful bacteria, and it helps and it enhance our digestive system. Okay, so that means that lactobacilli bacteria is good for human being, means it is good and useful for human welfare. So we are going to understand in this chapter or in this part about such useful bacteria only, or microorganisms. Okay, so now we're gonna start from the notes now. So microorganism benefits us in number of ways. Such microorganisms are called as useful microorganisms. Such microorganisms are called as useful microorganisms. Okay. Their uses can be divided into 4 categories, like where we can use that microorganism which are useful. We have divided those into 4 categories. So number 1 we have commercial level. Number 2 we have for medicinal purpose. Number 3 we have for agricultural purpose, and number 4 we have environmental protection or environmental. These are the 4 categories we have divided microorganisms on the basis of their uses. Okay, so let us discuss each one by one in detail. Okay, everyone. So first we are going to understand about the commercial uses. Okay. So what are the commercial uses of microorganisms? So some of the commercial uses of microorganisms are given below, and in which you are going to understand number one. Okay, for making curd and cheese. Making curd and cheese. So curd is being made at home. Whatever the process we have discussed here, like milk contains sugar, that sugar converted into lactic acid with the help of lactobacillus. So here we are using lactobacillus. Okay, so usually spoonful of the curd is added to the warm milk. So we have to go through the process. First we have the milk. Milk is being pasteurized, means we have to heat milk at a high temperature so that harmful bacteria may kill. Okay, and it will becomes warm. Okay, then warm milk we had to add some curd, like 1 teaspoon of curd, and we had to mix them well, okay, in a container, and we had to keep for 6 to 8 hours. So after 6 to 8 hours, what you will find, the milk has already been turned into the curd. So this is the process which turns into the curd overnight. So in a night, if you will observe that milk was turned into the curd. This happens because protein called casein present. See, very important. Which protein is present in milk? So it's casein. Okay, present in the milk coagulates. Okay, what is coagulation? To become thick and partially solid. Okay, and which is nothing but the curd beta. Okay, which is nothing but the curd. For casein to coagulate, the milk has to be made acidic. So for that, that milk have to be become acidic, and that acidic nature is being provided or maybe given to that milk is due to the lactobacillus bacteria. Okay, so the bacteria means the bacteria called lactobacillus. This is the name of the
Sir, I
bacteria
have a doubt, sir.
which is
Sir,
present. Yes,
casein
after
is an acid, sir.
discussion. No, beta. Casein is the protein present in the milk.
Okay, sir.
Yeah. So lactobacillus is nothing but the name of that bacteria which is present in milk. Present in the spoonful of the curd added to the milk. It converts the lactose. See, here we have the process that lactose sugar present in the milk turns into the lactic acid with the help of lactobacillus bacteria. This creates the acidic environment which is needed for the casein coagulation. Okay, so casein is the protein which is present in milk, which is being coagulated with the help of lactic acid. And lactic acid is formed by the lactobacillus bacteria from the lactose sugar. This process conversion of sugar into lactic acid. Just keep yourself mute condition, beta, everyone.
Sure.
Okay, the process of conversion of sugar into the acid or alcohol by the action of microorganism is called as fermentation. Very important. What is fermentation? Fermentation is the process in which lactic acid or alcohol is formed from the sugar, okay, or the hydrocarbon. This process is called as fermentation, and fermentation process takes place in absence of oxygen. That's why it is also called as anaerobic process. Okay, everyone. Okay, so now we are going to perform one activity. You can perform that activity at your home also. In this activity, we are going to prepare the curd from milk. Okay, what is the name of activity? To prepare curd from milk. So I'm writing here from milk. From milk. Okay, so first you have, what are the material we used to require? We require milk, obviously 1 glass, a teaspoon of curd, then a bowl and a lid to cover the bowl. Okay, what is the method for that? So ask an adult to warm the milk. Okay, so suppose here we have the container. Okay, here we have the container, and here we have to take the milk. Sorry. Then we had to warm that milk properly. Okay. And why we are applying this warming process to kill the harmful microorganism if they present in the milk. Okay, so to kill the harmful microorganism, we had to warm the milk first. Okay, after that, we had to transfer that milk into the bowl, another bowl. Suppose here we have another bowl. Here we have took that warm milk. Okay, like we had to wait some time so that temperature may reduce to warm or to warm, and then transfer the milk from bowl to a bowl and add a spoonful of the curd. So here we had to add spoonful of the curd. So this is the curd. We had to transfer that curd into the milk and we had to mix properly. Okay, we had to mix properly. Stir well and cover bowl with the lid, and we had to cover that bowl properly. Okay, with the help of lid, the cover. Okay, and leave the bowl undisturbed over a night, and we had to keep that bowl undisturbed overnight. Means it takes about 6 to 8 hours, but we had to keep that for 12 hours. And next day, what will be the result? The milk has transformed into the curd. So we will get curd next day. Okay. So what is the conclusion for that? The bacterium lactobacillus present in the spoonful of the curd added to the milk, which will turn milk into the curd. How? Because the lactose sugar which is present in milk is being converted into the lactic acid, and in presence of acidic medium, casein protein which is present in milk turns into like it undergoes coagulation, due to which the milk texture becomes very hard, and this is like hard and semi-solid. This is nothing but the curd. Okay, so note, adult supervision is required during the process. Adult supervision is required. Okay, so this is all about the activity. You can perform this activity at your home with your parents. Okay, so now we are going to understand which microorganism are going to use for the cheese and paneer formation. Okay, so the production of cheese and paneer, which is nothing but the cottage cheese, also involve the use of bacteria, okay, such as lactobacilli and streptococcus. Curd and cheese manufacturers also add a substance called rennet, and which is usually obtained from the stomach of the young cattle. Okay, the milk to make the process faster. Okay, why we are using that rennet? To milk to make the process faster. Whatever the process that we are applying to turn milk into the cheese or paneer, in order to perform that process fast, we are using that rennet. Okay. Addition of the rennet result in the formation of lactic acid. So once we will add rennet, the fermentation process takes place very fast, and which makes the milk more acidic. Okay. Next process we have making alcohol and beverages. So production of alcoholic beverages such as beer, wine, okay, it involves fermentation in which sugar present in the barleys and the grapes, which converts into the alcohol with the help of microscopic fungus called yeast. So if we will perform the fermentation process with the help of yeast, sugar converts into the alcohol. Okay, sugar converts into the alcohol and carbon dioxide is being released. Next process we have making bread. Okay, even for making bread we required microorganism. The process of making bread, which involves the kneading the mixture of flour, salt, sugar, yeast cells, and water into the dough. Okay, whatever the material we are using for the preparation of making bread, we had to form the dough first. Yeast converts that sugar to alcohol, and carbon dioxide is being released. As more and more carbon dioxide is produced during this process, that's why that dough rises in volume and forms the bread. Okay, this makes the bread porous. Suppose here we have the dough. If you will heat that dough in the furnace, that dough will convert or turns into the bread like this. Okay, because in bread we have many pore holes. In order to release the carbon dioxide, holes are being formed. That's why that bread and cake are very spongy. Even the dhokla we are used to prepare. Okay, they have like large number of holes in them. That's why this food becomes spongy. Okay, spongy. Baking of the expanded dough at 180 degrees Celsius kills the yeast and stop the fermentation. That's why whenever we are converting that dough into the bread, we are heating that dough in the furnace in which temperature is very high and where process of fermentation stops. The alcohol evaporates during the baking process. Okay, if suppose alcohol is being formed there, so it undergoes evaporation. Means in bread we have only the food, not the alcohol. Second last we have the fourth process, I think, making vinegar and coffee and tobacco by using microorganisms. So bacteria are used in the production of vinegar. Okay, that is nothing but the lactic acid. Oh, sorry, acetic acid. It has formula CH3COOH. It is beta acetic acid. Okay, it is also called as ethanoic acid. It's 4 to 6% aqueous solution of acetic acid is called as vinegar, and it is generally used in Chinese food. In Chinese food, tomato is not being used. So instead of tomato, to provide sour taste, they are using the vinegar. Okay. They are also used in processing the tea, coffee, and tobacco. Okay. So tea, coffee, and tobacco, they contain many drugs. So in order to remove most of the drugs, okay, from the tobacco, coffee, and tea, we are using microorganisms. We are treating these with the help of microorganisms. And removing caffeine from the coffee is called as decaffeination. Okay, next, last but not the least process, we have making toothpaste by using microorganisms. Shells of the diatoms, it is nothing but a kind of algae beta, okay, are used in toothpaste to give the gritty texture that helps the cleaning teeth. Okay, and the xanthan gum that we are using for the preparation of toothpaste, which is obtained from the bacterium called Xanthomonas campestris, and it is nothing but the name of the bacteria which forms the xanthan gum and which is used for making the toothpaste. It is also used in making toothpaste. So for in providing the dense texture to the toothpaste, we are using the xanthan gum, and which is being prepared by the microorganism. So this is all about the use of microorganisms in industrial level. Okay, everyone.